Morning. Let's get started. It's just gone 9:45. My name is Munipur Mohammed. I'm co-chairing this symposium with Professor Duncan Richards, who's from the University of Oxford. The symposium is called Toward an International Blueprint for Clinical Pharmacology. This is a symposium that's been organized by the Clinical Pharmacology Skills Alliance, which is an organization that has been formed by the British Pharmacological Society, the Association of British Pharmaceutical Industry, Faculty of Pharmaceutical Medicine, and Health Education England. The main aim of the Alliance was really to determine how we can work together across different sectors to promote the discipline of clinical pharmacology in its broadest sense in academia, in healthcare, in industry, and in regulation. So I was co-chair of that Alliance together with Dr. Andrew Foxley from AZ. So I'd like to thank Andrew for his help in preparing this symposium. I also want to thank Dr. Anna Zakaria, who was also involved in organizing the symposium but has recently left the British Pharmacological Society. So in this symposium, what we want to do is to explore how clinical pharmacology can help embrace complexity, new modalities, multi-morbidity polypharmacy, different sources of variability, and the interactions between them, so that we can make better decisions for all our patients who need the benefits across research, development, healthcare, and regulation. I think there's a need to explore the common principles and differences, discrepancies, through an international lens, and we've got people from different areas who are going to give talks today. And the golden thread that we're aiming for through the Skills Alliance really is, over the longer term, is for the diversity of populations we now encounter to be investigated with the diversity of modalities we have access to, with the aim of maximizing benefits and minimizing harms. And if you were here at the last talk from Mene Pengalos, you can see the many different modalities which are now available in order for us to be able to develop medicines and improve the use of existing medicines. So, with that introduction, what I want to do is first to introduce Professor Duncan Richards. Duncan is the Climax Professor of Clinical Therapeutics at the University of Oxford. Previously used to work at GSK. And Duncan is going to tell us about the future of early phase research from international landscape to the role of clinical pharmacology. Duncan, please. Thank you.
Thank you, Munir. Thank you, everyone, for the opportunity to speak to you today about a topic I think is especially important. You heard from Mene about the full scope of development of therapeutics from an industrial perspective. But I want to give you some thoughts now about the critical role that clinical pharmacology can play in the delivery of therapeutics for patients, and then focus particularly on early clinical development, where clinical pharmacologists have played an important role in the past, and I would suggest have an important role to play in the future. In terms of my declarations, I have a number of consultancies, although they're not directly relevant to what I'm going to be talking about today. I'm going to talk to you a little bit about some of the lessons we learned from the response to the COVID-19 pandemic, in which clinical pharmacologists were particularly prominent. And then I'm going to go on and identify some of the gaps that came from that response, and importantly, how we can take some of those forward as we think about developing novel therapeutics for the future, and some of the exciting developments, particularly in early clinical development, that we see coming forward. So, at the beginning of the COVID-19 pandemic, there was this enormous outpouring of enthusiasm for clinical research. And that's to be celebrated. However, there were some problems. Peer review takes time, oversight takes time, and many of those things went out of the window, frankly. And everyone was driven, quite properly, by a need to move at pace. One of the downsides of that was there was substantial, what I would describe as political involvement in the selection of which trials went forward. And I don't necessarily mean just by politicians, but within the medical community. There was quite a lot of influence by senior people of what went forward, and that wasn't always based upon the strongest science. And what we ended up with was a very large number of phase 2 studies that were frankly not powered. Many of them used the WHO scale, and frankly there is nothing wrong with the WHO scale, but it is a phase 3 tool really, because it requires 200 to 400 subjects per arm in order to be able to detect a meaningful difference. And you have to ask the question, the differences of one score on this scale are quite different depending on where you are on that scale. And so what we ended up with was a very large number of underpowered studies, which did not lead to practice-changing research. And regardless of what you thought about hydroxychloroquine, the last thing we need is 290 trials looking at it. So that's what we saw. What did we really want to see? Well, what you really want to see in this sort of rapid response mode is seamless integration between the different phases of research, each one feeding off and handing over to the next, generating the data that is needed to be able to move rapidly between these phases. Inevitably, you're going to be starting with repurposing of drugs, but you also want to see the newer and exciting drugs being brought forward, generating that data quickly so that you can change practice quickly. Well, did we do that? Well, what actually happened was, yes, there was lots of repurposing, but a lot of that repurposing was based on secondary and quite often poorly defined secondary pharmacology. People used the standard clinical doses that were used for their primary indication, not often linked to the pharmacology that they were wanting to engage. And there was very, very little attention to exposure response in many of these studies. And if you look at the proportion of reported trials that actually generated actionable data in the COVID response, it's less than 5%. So that's a description of the problem. How could we address that? And I would suggest that as the response to the pandemic evolved, we did see some important changes in the way that things were structured to improve the throughput of trials and of results in order to be able to inform clinical practice. And I would suggest to you that clinical pharmacologists were pivotal in that. So our chief scientist in the UK was advising government, and he is a clinical pharmacologist. We have important people such as Munir, who were advising the branch of government responsible for regulation, the MHRA. We had clinical pharmacologists prominent on UK CTAP, which I'll go on to describe in a moment, which was an integrated way to try to think about therapeutic strategy in this space, and also to then inform the major funders of the large platform studies that were prominent in the response to COVID-19. And in each of the large platform studies, many of which will be familiar to you, clinical pharmacologists were prominent in both the designing, execution, and interpretation of those studies. So as a discipline, clinical pharmacology stepped up to the mark in COVID-19, and I think many of its contributions were extremely positive in turning what was quite a chaotic landscape into something that was more organized. Well, what did that landscape actually go on to deliver? What this describes is the large major platform studies that were running in the UK at the time, many of which we very familiar with to you, but some perhaps less so. And there are some important lessons here. Although many of these studies have delivered answers and actionable answers, you'll see at the bottom that there are the blue section, which are things that are still running for which we don't have answers. And despite the scale of the effort, despite the number of patients infected with this disease, we've still got a large number of sectors for which we don't have good evidence about what to do. And as many of you will know, the opportunity to recruit patients into clinical trials now is substantially dwindling, and therefore many of these questions may remain unanswered. And it's an important principle that despite the large numbers of patients, despite the very large numbers of patients who were enrolled into clinical trials, the amount of actionable data was small, and we need to think systematically both about our therapeutic strategy in these situations, but also about ensuring that patients are enrolled into studies that can generate actionable data. I want to take a specific example now of phase 2. So in the ideal situation, phase 2 studies are generating information on candidate drugs which give an indication of whether this is a drug that could, in a larger phase 3 clinical endpoint study, be beneficial for patients. The idea is that it should be small and it should be quick, so that you can then feed those things through into the large platform studies. That isn't actually what happened in COVID-19. It was actually the phase 3 studies that were set up first and were generating things more quickly. And one thing we didn't do a good job on was really coming up with phase 2 platforms that were quick and able to deliver actionable information into phase 3. And I want to give you a specific example that I was involved in. There were 3 national platform studies in the UK, and as of today, this is the only one that's got published data. So I think that's an illustration of some of the challenges that were faced by people working in this space. We selected CRP as our surrogate endpoint. Now, we can debate at length, and I'm very happy to talk to people at length afterwards about the selection of that particular endpoint, but the point is CRP is a phase 2 type endpoint, i.e. it is a surrogate biological marker that we believe is linked to therapeutic response, but is not that therapeutic response. You need a much larger study to do that. And we looked at 2 things, both of which had a reasonable biological hypothesis to support them, anti-GM-CSF and infliximab. And we ran this study, and we were able to demonstrate that the response to anti-GM-CSF was positive, and the response to anti-TNF was not positive. And therefore, a recommendation, and this was accepted, was that anti-GM-CSF should be tested in phase 3. Because of the time that it took to do that, that test has never actually taken place. So we've never actually been able to test whether what we did was right or not. But subsequent to this, some data has come out from the biological community looking at samples from patients with COVID. And if you look at the complex work, the omic portfolio here, that there's pretty good evidence from the combat data that does suggest that anti-GM-CSF should have worked and that actually anti-TNF was unlikely to work. So again, we've done all this in the wrong order. In the ideal world, we would have done this piece of work first to give us a signal for those drugs that we ought to have tested. We should have done Catalyst quickly. Just as a reminder, the number of subjects per arm here is much more appropriate to phase 2 response. And then we should have been able to take the winners into the large platform studies such as recovery. The other key piece for me is thinking about therapeutic strategy. Again, what we saw in the COVID-19 response was quite a lot of drugs being tested in the patient population that people had greatest access to, without necessarily thinking through whether that intervention was best suited to the evolution of the disease at that point. The other point that I've also made was despite the very large numbers of patients involved, we cannot test everything, and therefore it is really important that we pick carefully those things that are most important. And the UK CTAP was a response to that, because it was frankly a bit of a zoo at the moment when things began in terms of selecting which drugs were going to be tested going forward. And so UK CTAP was set up under the auspices of UKRI to provide an independent secretariat that would provide dispassionate reviews of proposed targets, but alongside that would also pay attention to exposure response and identify whether there were doses available that could engage the proposed pharmacology. Would also think about the drug supply, because if there's only 2 grams of this drug in the world, even if we generate a positive response, it's not going to be a useful treatment for patients in the near future. But to take this broader approach, and also to think about where in the therapeutic pathway this drug was most likely to be impactful, and therefore to recommend it to go into the individual platforms. And importantly, the committee was properly constituted with proper declarations of interests. It was clear and minuted what the decisions were and the basis for those decisions, which again provide a level of confidence that that decision making was appropriate. And this is a piece of work that I'm very proud to have been involved with. The team produced, and it was only a small team of 6 or 8 people, over 100 of these briefing documents, which were often extremely detailed and very high quality, providing dispassionate and clear information on what the real data was drawn from across the literature, plus the information provided by the proposed sponsors of these informations. And we met very frequently in order to review these things, in order to not hold things up, and made a number of recommendations into the national platforms. But again, because of the amount of time that it took to set this up, to decide that it was needed, set it up, and then actually do it, the proportion of those that actually went on to be tested is relatively small. So thinking about the future, this is a framework that we should think about for the future. And I'm very pleased to say that a number of the new platforms that are proposed for looking at common diseases which are learning from the experience of COVID-19 have all taken on board the idea of having a therapeutics strategy committee alongside their main trial management committee to think about the interventions that should be tested within that. And a lot of that is based upon the success of UK CTAP, and I think that is very positive. So just to sum up this section, inevitably you're going to be repurposing phase 3 drugs in the first instance. We then see a phase where you're repurposing new drugs. So these are drugs that are still in development, but you're using them to see, and typically these were directly acting antivirals to see whether they were relevant for COVID. And now we're beginning to see new discovery assets coming through. The challenge, however, is how we're going to be able to test those given the environment we're now in. And I think clinical pharmacology had and continues to have an important role to play. And importantly, there are some lessons here that we need to think about for any future response. It's really great that everybody was really enthusiastic about doing research, but if you're going to do actionable research, that does need to be coordinated both at a national and at a local level. And clinical pharmacologists, I would suggest, are particularly well suited to do that because the training is across the therapeutic landscape, not in a specific organ specialty, and therefore it allows people to have a broader view, which is particularly necessary in these situations. Even in a pandemic, you can't test everything, and therefore judicious choices have to be made, and those need to be made on good understanding of the pharmacology that you are testing, and not just repurposing existing dosages with unknown pharmacology. A lot of patients were involved in studies that really had no realistic opportunity to deliver benefit, and that isn't to the benefit of everybody. There's quite a lot of questions that we still haven't answered, and a lot of these fall firmly in the clin-pharm arena. Combination therapy. We normally treat viral diseases with combination therapy. And to think about how we develop the host-directed antivirals, because we haven't really got very much in that space at the moment. So that, to me, gives us a view of clinical pharmacology, but I'm an early clinical development person, and as I've highlighted, our response in the COVID-19 in phase 2 was not one of our strongest points. How can we improve things in the future, and how do we think about clinical pharmacologists playing a critical role? If we look back in the past, the golden age of clinical pharmacology was often based upon direct physiological measures, blood pressure, gastric pH, these types of things. And the critical point about that was, this addresses the point that Mene was making earlier in his lecture, which was, these are direct measures of the pharmacology that you're interested in, and therefore the translational value of that information is extremely high. We then moved on to a long period which was rather less successful, where we've particularly focused on biomarkers, and particularly those associated, those in the blood. And there are some successes here, but there's an awful lot of failures, because frankly, the blood is soaked in the drug, and therefore engaging the target is not particularly difficult in that space. And many of the markers have proven not to be predictive of the future. So while I think there will continue to be an important role for this, we need to take a more sophisticated approach, and we need to think about tissue. And I think this is the real opportunity for clinical pharmacology in the early clinical phase going forward. Because as has already been presented, phase 2 really does remain the valley of death. For the development of new drugs. These are the data for 2016, and then some from the Pfizer portfolio from more recently. And what you'll see is that the overall success rate, yes, and Pfizer were congratulating themselves quite appropriately that they've improved their success rate, as AstraZeneca seem to have been able to do, from a pretty paltry 8% up to the best part of 20%. But that still means 4 out of 5 things are failing, and the principal area where failure is taking place is in phase 2. And as has been mentioned, these days, that is because the drug doesn't do as much as we wanted it to, or doesn't do anything at all, rather than that it is toxic or has adverse pharmacokinetic properties. So what is the situation that we find ourselves in today? And this is a very busy slide, and I'm not expecting you to read it, but it just illustrates a recent review looking at targets for inflammatory bowel disease. And in common with most of the diseases that we work on, there is no shortage of targets these days, but these maps do not tell you which are the ones that don't have substantial redundancy, and don't tell you which are the subgroups of patients that are likely to have substantial benefit associated with them. So how can we do a better job of identifying that? And there are various systems. This is the 3 pillars which came out of the Pfizer portfolio. We heard earlier about the AstraZeneca's 5Rs. They're all versions of the same thing. And basically, they're asking some fairly simple questions, not always questions that are easy to answer. But I want to focus particularly on that pillar 3, because this is the crucial one. Because to me, all too often, we are focusing pillar 3 not very differently from Pillar 1, in that many of our markers of pharmacology are really very proximal measures of pharmacology, which are little more, frankly, than measures of target engagement. And what we really want, and if we go back to the history, the whole point about measuring blood pressure is that is an integrated measure of the effect of your drug, and actually gives you a high level of confidence that the effects that you measure are relevant. And I just illustrate this, that we're actually pretty good at pillars 1 and 2, but quite often pillar 3 is a bit wonky. What can we do to improve that? And I think the real opportunity in this space now is to harness the power of the Human Cell Atlas. This is a global initiative to really understand the cellular basis of disease. Our phenotypic classifications of disease are pretty hopeless, and they don't really tell us the underlying pathology that is in fact the driver of the disease, and therefore the driver of the opportunities for therapeutics. And there are lots of examples of this, but this is just one from my own university, where we are working in this case with Janssen to do very detailed profiling of the patient populations of interest to really understand what are the cellular players, what are they producing, which ones are active, and at what stage of the disease. Genetics is very, very important, but genetics doesn't tell you that that particular pathway is active at the point that you are going to intervene. And there have been a number of failures of very well genetically validated targets, because probably it was important at some other point. And where does this lead to? Well, I think this leads to the basket trial, where we identify disease states of interest, but we ask a biological question, a cellular-based question, which says, in these 3 disease states, which I have pre-identified as being of particular interest, when I intervene with my target using cellular-based endpoints, which is the disease state where I really see engagement of that pharmacology and a movement of the biological endpoint in a way that I have predicted would potentially be beneficial. And this should allow us to be more clear in our selection of patient populations, but also to improve the translational predictivity of what we do when we then go into a properly powered phase 2b study with proper clinical endpoints, rather than the plethora of rather small and underpowered studies that have been a particular feature of drug development in the last 10 years or so, which we know are associated with a high risk of failure in phase 3, which is both expensive but also a tremendous waste of effort on both patients and researchers' behalves. I just want to finish by highlighting that this is not the only approach. And for a long time, we have used infection challenges as a way of evaluating vaccines. But I would suggest that that has been somewhat siloed in the vaccine world. But if we think about it from first principles, in essence, we are stimulating the immune system in a way, and we are looking for a response. And there is much, I think, that we can learn from those infection challenge studies and think about them in the context of something that my group is particularly interested in, which is the use of immune challenge models as a way of interrogating drug action at the very earliest stages of human clinical development, phase 1b, really being able to interrogate how a new drug target is modulating the immune system so that we can think more sensibly about indication selection and which are the disease states that we should evaluate in our basket trial. And I think this is a real opportunity. But the landscape at the moment is that most of these immune challenges have not been optimized for drug development. And if we just take the most famous example, lipopolysaccharide, which has been given to humans for 20, 30 years now, it's got a very narrow therapeutic index, well, not therapeutic, in a very narrow tolerability index, rather. And you can induce a sepsis-like syndrome with 4 nanograms per kilogram, but your volunteers tend not to come back under those circumstances. And that's not really what we're wanting to do. This is not a disease model. We are wanting to stimulate the immune system in a predictable and reproducible way, not to pretend that we're inducing some disease state and looking at that. And so we need to, and if you look at the responses here, just of those different dose ranges, there's very little, the responses are highly variable, and there's very little clear blue water between these things. So this is not the good basis for a good reproducible immune challenge. And so we need to think about things differently. And my team are particularly working on the use of keyhole limpet hemocyanin, which is a neoantigen approach, for which there is already quite a decent amount of data in the literature, but there isn't that information on the details of dose response and the optimal way of measuring these things. And just as a specific example of this, which is again something that came up in COVID, a lot of our measures are optical based, and it matters what skin color you have. A lot of the measures work very well on light colored skin. They don't work very well on darker colored skin. And if we're going to do clinical development properly for everybody, we need measures that are appropriate across the different patient groups. So I want to just finish with two points. One was there was a lot that was very positive about the response to COVID-19, but there has identified some important gaps. But I would suggest that the skills of clinical pharmacologists can be very important in shaping and understanding our responses and taking that forward, particularly in therapeutic strategy and early development. And I would also suggest that there was a golden phase of clinical pharmacology in drug development when we were looking at physiological measures. I think things went off a bit when we focused entirely on biomarkers, but there's a real opportunity in the future to harness the human cell atlas and really use that as a cellular basis disease, and clinical pharmacologists should be at the heart of that. My work involves lots of different teams, and I'd just like to acknowledge them on the last slide. And thanks very much, and I'll take any questions you have.
Thank you very much, Duncan. Any questions? Emma.
Duncan, thanks. It's Emma Baker from London. A lovely talk, and I was there in the pandemic recruiting patients to these trials. We had a sort of backwards design, didn't we, in COVID? And one of the problems was that we couldn't recruit to phase 2 trials because everyone wanted to be in the phase 3 trials. What's the role of clinical pharmacology, do you think, in that message about the importance of phase 2 trials?
So this for me was part of the local response. So one of the things that we did in Oxford was put together a multidisciplinary group of all of the people who were involved in the clinical trials and came to an agreement about the order in which patients are going to be offered clinical trials, because you absolutely cannot have a stampede of people approaching a very sick patient to offer them a range of different studies and have them try to decide that. You need to do that. And that's a local response piece. But as you say, what we didn't do at a national level was really come to an agreement about where we were going to focus our efforts in terms of where the phase 2 studies were going to be done. Because frankly, you have to do them at, they have to be offered preferentially over the generally much simpler and much easier to run phase 3 studies. And so we didn't ever really get that agreement. It was all done on a sort of local basis, and I would say that that was quite patchy. I would hope in the future that there would be an understanding that actually you need to enable the entire pipeline. I think, the agile phase 1 platform has been successful and generated a number of important results. But the way they did that was to pull it out into some different environments, which meant that they weren't then competing in the melee of the general hospital.
Thank you.
Okay, we've got a question at the back from Jeff Aronson, and then there's somebody at microphone 4.
Thank you, Jeff Aronson, Oxford. Thanks, Duncan, for a very clear and pertinent lecture. You mentioned repurposing, and I think that there are different types of repurposing. We have the variety in which you take an existing drug with a known mechanism of action, known to be effective in some other condition, and you use it for that mechanism in the new condition. Dexamethasone, immunosuppressant here, immunosuppressant there, it works. Great. Clinical trial. The other type is less successful. You take a drug that is known to be effective in some condition with some mechanism, and you use it for the new condition, but under the supposition that it has some other mechanism of action, something that isn't supported by your 3 pillars, which incidentally is very similar and mechanistic to the IDOS classification of adverse drug reactions. Does the drug hit the target? What does it do when it gets there? Does that translate? These drugs like hydroxychloroquine, ivermectin, failed that test. Now, I think as clinical pharmacologists, we should be promoting more vigorously mechanistic reasoning, which quite a lot of people seem to think is not helpful, largely because of a small number of egregious examples that show that the mechanism doesn't necessarily link to outcome. They're very few. In the majority of cases, mechanism is hugely important, and I think we're not stressing that enough.
I would agree entirely. If you look at the way that we are developing new medicines, it just seems to work. I don't know why, but it seems to work. Let's carry on approach. That's the classic phase 2 response that then fails in phase 3 after you've spent another £400 million on development and wasted a lot of time and money. So I agree entirely, and to me that was the critical thing that the CTAP approach took, was there was an entire selection on exposure response, which was, I would say, of the things that was always missing from the submissions, it was a clear description of how exposure response was going to be relevant with the repurposed drug that was being used.
Number 4.
Thank you, Professor. I'm Ted from Malaysia. I just want to know, first of all, thank you for your insightful presentation for us. Regarding the 3 pillars, the third one, the evidence of pharmacology, can we consider evidence-based medicine, network meta-analysis, systematic review as a part of the third pillar, or should it be the 4th pillar? Because I noticed that there was one Cochrane review during the COVID-19 regarding the chloroquine use, and at their conclusion, it even mentioned that no further trial of chloroquine on COVID-19 patient. It does not recommend chloroquine, as well as it stopped the trial, begging to stop the trial. So I think evidence-based medicine, could it be the part of the pillar, or what's your advice, Prof? Thank you.
So I think they're 2 slightly different things. The 3 pillars are directly relevant where you're developing something new, where you've got existing evidence. And the Cochrane review is one. There are plenty of other reviews that are very clear that hydroxychloroquine is never going to do the thing that you think you want it to do, because you're not going to be able to engage the pharmacology that you think you're going to engage. So to me, in some ways, you don't need the 3 pillars. It falls over on all the 3 pillars to start with. What the Cochrane review is provide you with the evidence to do that. The other crucial piece is you have to pay attention to that evidence, and there was plenty of examples of people not paying attention to evidence in COVID-19.
Thank you, Prof.
Okay, we've got 2 more questions. Robin Ferner, and then microphone 1, please.
Yes, thank you very much, Duncan, for an excellent lecture. Do you have a solution to the problem of treating orphan diseases? So
to me, By orphan diseases, you mean rare diseases, or do you mean underserved, neglected diseases?
I mean rare diseases,
Rare disease.
where phase 3 trials are difficult or impossible.
So rare disease is quite a broad spectrum. You have things like pulmonary arterial hypertension, which, albeit rare, there are sufficient subjects that you can do properly powered phase 3 studies. And then you have ultra-orphan things, where you're literally talking about a handful of patients. My personal view is the 3 pillars still apply, and if rigorously applied, give us the evidence base to then think about how we then take things forward. And that's where we have to think in partnership with the regulators about what the necessary level of evidence will be to get to take those things forward. And we do need to think about there are structures in place around conditional approval, etc. The issue for me is we haven't really seen we've seen the carrot used very extensively. We haven't really seen the stick used very extensively. And that, for me, feels more like a political problem than necessarily a failure of the regulatory infrastructure. It's the way that we apply it.
So for Munia rather than for you, perhaps.
Okay, number 1, please.
Good morning, Vimal here from South Africa. Thank you for your great presentation. You gave us a lot of insight. My question is on, do you think that there is a need for some sort of plan or sort of framework with which we can maybe recommend to either the World Health Organization or different maybe government organizations how to prepare in the event of further or future pandemics, so that, I mean, you know, case in point was when you mentioned 289 clinical trials or trials on, I think it was, I can't remember the drug itself, but do you think that there is a need for some sort of framework to work on something like this, to prevent repetitive work?
So the answer is yes. The truth is, however, there are umpteen plans which are sitting in filing cabinets around the world, which were generated after previous disasters, which didn't get used this time, just like every other time they didn't get used. So for me, it's about thinking about how do we have structures in place. And I know my colleagues like Peter Horby are thinking about how do we have an infrastructure that is running and doing useful work that can then pivot when things are, when the need arises. If you look at things like REMAP-CAP and ISARIC, they were already up and running and working. They then pivoted very rapidly to do useful work in the pandemic. It's very hard to set up other things very quickly, but also nobody wants to pay for large infrastructure that is sitting around just in case we need it. And actually it doesn't really work, because what do you do with having 100 people just in case? So I think we need to think about how we organize ourselves with structures that can work in the inter-pandemic periods, but then can rapidly pivot.
Thank you.
Great. Thank you very much, Duncan.
Thank you.
Okay, great pleasure to introduce the second speaker. This is Dr. Alex Phipps, who is a clinical pharmacologist at AstraZeneca at the moment, leads the oncology portfolio, the clinical pharmacology of the oncology portfolio at AstraZeneca, previously at Pfizer and Roche. And thanks very much, Alex, for agreeing to do this talk.
Thank you, Mene. Good morning, everyone. I'd like to thank the organizers for the invitation, and pleasure to be here today to give you a perspective on the future of systems pharmacology. So my disclaimers: I am an employee of AstraZeneca and a stockholder, but the views expressed in this presentation are my own and not necessarily reflective of the company. So really what I want to do I guess another disclaimer is I'm not a systems pharmacologist. I have a pharmacometric and clinical pharmacology background, but I lead a group whose work is heavily influenced by the systems pharmacology which is produced within our organization to help us make better decisions in the clinical development programs. So it's my perspective on where that is at the moment, where it goes in the future, and how it helps us create better medicines in the future. I'm going to do that by talking a bit about the status of systems pharmacology today, what are the trends for progression in the future. Then I want to highlight 3 specific examples, applied examples. I won't go into too much technical detail, but I really want to make some points about those examples because I think they really offer things in terms of offering mechanistic insight in the case of diabetes, unearthing patient variability in response. This is in the context of immunotherapy, but I think there's some profound learnings in this example. And then providing informed dosing, scheduling adjustment in the treatment of cytokine release syndrome. And we just heard about COVID and cytokine release syndrome, something there as well as in the immunotherapy area. Then I want to consider future developments and refinements in systems pharmacology. So I use this, I've just noticed I have a pointer as well. I use this standard sort of basic overview of a QSP modeling approach where we have the features of the pharmacology, the dose, the regimen, the potency of the molecule. We combine with the features of the biology, the target of the therapy, downstream effectors, molecular, cellular, etc., that multi-scale modeling, which can be quite problematic to fix. And then finally, the physiologically based endpoint, a classic being, of course, longitudinal tumor size in oncology. I did a quick search through PubMed and found, I didn't go through all 3,910, but there were almost 4,000 publications on QSP modeling over the last 20 years. And there's a steady stream of 300 to 400 per year. So this is a very productive area, especially when you think of the potential complexity for each of these publications as well. And they're across multiple disease areas. So I don't think there are particularly disease areas that are underserved by these approaches. Multiple modalities and at different stages of the sort of basic science, academic research, drug research and development. So, where can we make further progress? I think we can still aim to get a greater understanding of the pathophysiology underwriting the mechanism of action of the drug. We can improve our quantification and understanding of drug variability. As I say, I'll highlight that. But also, what drives that? And can we harness that in our drug design? Can we combine separate models into a single QSP model, bolting one onto another to get a greater in-depth understanding? And that's really where machine learning, artificial intelligence comes in, to be able to deal with that amalgamation of a large data set and turn it into something meaningful and easily understandable. Right, talk about the first example. And this is a modeling approach to understand the physiological biochemical cascade and also providing pharmacological insight into meta-analysis across large clinical trials. And this is a QSP framework which was developed to enable efficacy and safety differentiation between SGLT inhibitors, the glyphosins, so the sodium glucose transporters, in type 2 diabetes mellitus. The work is by Gabriel Helmlinger, etc., et al. So, though I'm sure most people in the audience know that SGLT inhibitors are an approved class of anti-diabetic medications. They either hit SGLT1 or SGLT2, and they inhibit glucose reabsorption in the kidney, leading to a reduction in plasma glucose, increased urinary glucose excretion, and a lowering of HbA1c. To compare glyphosins requires large clinical studies and meta-analyses, so it's not easy to gain that insight and determine which are the best for the patient. And this QSP model was built to incorporate mechanistic, pharmacological, and physiological explanations for similarities and differences between the glyphosin class of molecules. So on the left-hand side here, I'm conscious if I turn my head, I lose the microphone. This is a model of the kidney, healthy and diabetic kidney. And what you can see on this is the proximal tubule moving into the bladder with UGD being urinary glucose excretion. You'll see a number of lines on here, so some of them are drug and glucose fluxes, glucose reabsorption back into the plasma as well. Inhibition by the glyphosins, which are the red boxes, so they're inhibiting the SGLT2 and SGLT1. And the parameters in red are fitted parameters from our knowledge of glyphosins The parameters in green are parameters about kinetics of reabsorption of glucose from the literature. So the results showed, firstly, they could simulate the results from a hyperglycemic clamp experiment in healthy volunteers and diabetic patients that showed predicting maximal SGLT1 and 2 contribution to glucose reabsorption. You can see on the left-hand side that the SGLT2 is a far major contributor. But then on the right-hand side, what I like about this is a couple of things were counterintuitive that the model unveils. So on the top 4 figures are drug concentrations in plasma, in the S1-2 proximal tubules, S3, and in the bladder. And I had intuitively thought, well, the concentration is going to be similar, but it's not. Whilst the time courses are the same, there are different concentrations across the different components of the tubule. But what we see on the bottom is the inhibition of the inhibition of the total reabsorption on the left, the inhibition of the SGLT2 contribution, as expected, the drug is working, but there's a counter SGLT1 contribution in the context of dapagliflozin, and then finally a cumulative increase in urinary glucose excretion, showing the drug is working. But this gives insight into a dynamic shift that appears to occur with SGLT1, SGLT2 specificity, and resolves an apparent paradox where apparently the contribution to clearance of SGLT2 is about 80% in healthy volunteers, although only 30 to 50% in diabetic patients. So because of that, so SGLT1 also produces very high level of inhibition of glucose reabsorption, but it can have side effects. As a consequence, it can lead to glucosuria and mycotic infection risk, urinary tract infection risks. So there's a benefit-risk balance here. So there'd been a meta-analysis undertaken across multiple studies and had just not actually looked at urinary glucose excretion. So what this model did was produce a simulated control arm on the UGG, urinary glucose excretion levels across the different glyphosins. And they predicted that there was a 2 to 14-fold increase in glucosuria and downstream side effects with those less selective SGLT1-2 inhibitors. So this shows that by adding that QSP, you can get a better understanding of actually what that clinical meta-analysis is telling you and the relevance for patients. I want to talk a bit about hypothetical variability in drug efficacy and safety, and this comes from Richard Peck's seminar in ClinPharm Therapeutics in 2020. And basically what it's showing is the green lines are the dose where the patient's deriving benefit, the red is where they'll derive a side effect. And you can see in a wide therapeutic window, there's no problem there, the dose sits in the middle. In a no population therapeutic window, it's all over the place, it's all intertwined. And then a narrow therapeutic window, something like warfarin, perhaps here, you've got a very narrow difference between the benefit and the risk. But how large is that variability? You notice there's no numbers on the y-axis here. And we also know that that variability exists at many stages between dose and effect. So the PK/PD will give us insight into the sort of dose, plasma concentration, effect site concentration. The variability in the interaction with the molecular target, interaction with disease, biochemical cascade, etc., that's where the QSP model comes in. So I want to move on to a model which has just been published at AACR, so American Association of Cancer Research, so it's not actually a full manuscript yet. It's by Christina Santini et al. From Rush. And what they did was looked at a QSP model to understand varying response and safety and to optimize dose and schedule in real time. And this is with a T-cell engager. I'm going to explain what a T-cell engager is to those in the audience who may not be familiar. But this T-cell engager is targeting the antigen GPRC5D in relapsed refractory multiple myeloma. So in this figure, what you can see is an antibody which is binding to the target antigen in the myeloma cell. And then the second arm of the antibody binds to the CD3 antigen, it could also be a CD8 antigen, of a T cell. So effectively, it's recruiting the T cell via antigen presentation between the T cell and the myeloma cell, and induces activation of the T cell and killing of the cancer cell. That formation between the antibody, the myeloma cell and the tumor cell, and the T cell, is called a trimer. And I will refer back to trimer and trimer concentration throughout the course of the presentation. As a consequence of that immune cell activation, though, the immune cells release cytokines. And this is where cytokine release comes in. So with chimeric antigen receptor T cells or with T cell engagers, cytokine release is a major concern and difficult to manage, and it's led to sort of, particularly with T cell engagers, routes of step-up dosing where you start at a lower dose and dose escalate. But that's been really rather arbitrary. So the modeling strategy with this work was to perform patient-specific calibration, then to characterize the population with regard to patient sensitivity, to tumor killing and immune activation. Is there any difference between the two? Can you separate the two from each other? And then to characterize associated heterogeneity. Then they simulated different dosing regimens in virtual populations to predict the probability of both response and CRS, i.e., can we maximize that therapeutic window? And what I really find most elegant about this model is that it's simple. So many QSP models take a page with lots of different figures, lots of different parameters, lots of different equations. This one has 15 parameters. It's a simple, ordinary differential equation sequence. And what they do is that in silico, they use the soluble B-cell maturation antigen, SBCMA, as a surrogate for tumor burden. So you can monitor that in the clinic. And it informs on the probability of response. And you can derive an EC50 for that tumor killing. They also look at the maximum release of interleukin-8 as a surrogate for immune activation. Surprised me that. Normally we see IL-6 or TNF-alpha or interferon gamma or CXCL10. This one used IL-8. And it informs on the probability of CRS. And you can derive an EC50 for immune activation. And then it's validated with clinical data in phase 1 myeloma patients. And the model predicts the clinical data well. So on the top, we see a predicted reduction in the soluble BCMA. In the middle, this is a simulation of activation of proliferation of peripheral T cells. And finally, they predict the changes in interleukin-8 with dose. Noteworthy, you see that the interleukin-8 levels go down with time as the system starts to tolerate that immune activation. And my final example will give some insight into why that happens. So, this is why I put this figure up more than anything, because what we see is that Santini et al. Derived an EC50 for tumor killing. So on this figure, the red dots are those patients who have not responded, and the green dots are those patients who have responded. And what you can see is on the axis, you can see a clear differentiation, a relationship in EC50 tumor killing to patient response on the left-hand side. Whereas the EC50 to immune activation appears to overlap. There's no clear association. That means potentially that all patients are going to get CRS, but not all those patients are going to respond. There's a 95% accuracy in predicting that partial response or better, and 80% accuracy in predicting CRS after first step-up dose. I would submit to the audience that with a phase 1 trial, that's pretty impressive in terms of being confident about simulating doses and schedules. But look at that y-axis. There's a 4 log unit variability in the ability to kill the tumor cells. That is your patient variability. And the consequence of how we deal with that, how we predict that, where an individual patient may be, that's where I think the strength of potentially with QSP, with artificial intelligence, with omics data, single-cell sequencing, et cetera, will give us a better understanding of where any individual patient may be and whether this is surmountable for all patients or not. I can't imagine it is. 4 log units of dose is clearly not going to be achievable in the clinic. But this really showed insight to me. So in the context of combining system model, I want to come on to one final example before I go on to my sort of prospectus for the future. And this is staying in this same theme with T-cell engagers. I do have a bias, having worked in oncology for a number of years, but this I found a particularly profound piece of work. And actually, the author, Jared Weddle from Astellas, presented at a webinar a few weeks ago, so I'd already written this in my deck, so I hope I can do it justice to the brilliance of his work. So this is with the T-cell engager tebentafusp, which is the first approved T-cell engager in solid tumor context. There are 4 approved in hematology setting. And it also binds to GP100, presented in the context of MHC class 1 on the tumor cell. So it's not a specific protein antigen, it's a peptide fragment expressed as a consequence of tumor cell activity, so it's deemed to be more specific. However, GP100 is also expressed in healthy skin tissue as well. High levels of cytokine release were observed, resulting in alternative dosing strategies. The cytokine release observed was in 85% of patients in the first cycles of dosing. So what Dr. Weddell did was consider, how does this happen? And so what you're going to see here is the most technical slide I'm going to present. And there's no equations, but we have a 4-compartment model. And the model structure looked at 5 cytokines. They actually looked at 15, but only presented 5 of them finally, just for, I think, for brevity, although there is a large supplementary index with the manuscript. Those cytokines being interferon gamma, interleukin 6, interleukin 10, and CXCL10 and 11. One of the assumptions in the model is that the GP100 expression in the melanocyte, the healthy melanocyte, is 1/100th of the tumor expression of the target antigen. So on the left-hand side, we have peripheral and central compartments, which is antibody kinetics, and also the kinetics of released cytokines. But on the right-hand side, there's a tumor compartment, and below it, there is the healthy skin compartment. But in both stages, the formation of a trimer will lead to activation not only of the T cells which are engaged, but non-secreting immune cells, which could be monocytes, they could be neutrophils, they could be NK cells, will transition into a secreting state and release cytokines. And then they will then transition into a refractory state. There are 3 states of those cells. There's fast transitions of non-secreting to secreting and secreting to refractory, with a slow transition from refractory to non-secreting, which helps to explain why we see this tolerance, that those activatable cells are in a refractory state after the first or second doses. So what do the results look like? Firstly, the model predicted the profiles of each cytokine. You can see with dose, so this is what I'm talking about, this tolerance or tachyphylaxis, that the levels of cytokine changes go down with time. But the crucial point is, what are the side effects associated with those highest levels? Part A here shows the formation of that trimer, which is a surrogate for efficacy. And you can see that it's about the same with time. And actually, when they simulate it across doses, so if you do a step-up dosing, you still get to the maximum concentration of trimer, but you will reduce the level of cytokine release at those earlier stages. That's critical. For me, that's the first proof that actually step-up dosing works, that you don't ablate your efficacy by doing this cautious dose escalation approach. Now, on the right-hand side here, B, C, and D show the transition state. So we can see the reduction in the non-secreting cells and the secreting cells with time, and the increase in cells in a refractory state. So there's a beautiful simulation of changes in states. Of course, these are not identifiable necessarily as obvious cellular states. One thing the model couldn't identify was the conversion time for refractory cells back to non-secreting cells, because it appeared to take too long, and it wasn't identifiable. But I think that's important as well. And then they looked at the differences in different dose optimization. So on the top, what I'm showing is different simulations of dose escalation, step-up dosing. So you might start at 10 micrograms, you might start at 56 micrograms. So you can see the higher your first dose, the higher the cytokine levels, and then they go down. But if you encroach upon that optimal dose, you reduce the extremity of the cytokine release. But there's a trade-off here, because in those first cycles, or particularly on the first cycle, the patients are hospitalized. They're monitored. If they present with cytokine-like syndromes, they can be treated. If the patients are effectively, the step-up dosing is kicking the cytokine release down the road. They could be going into hospital, receiving their infusion, going back home, and then the cytokine release could occur in a home setting, harder to treat as a consequence of that as well. But what it shows is you can kind of identify a sweet spot here. And finally, they looked at a sensitivity analysis. So they varied across fourfold parameter sensitivity. And what they found was actually the pharmacokinetics of the drug wasn't the key to the only key to the cytokine release. They found it was actually more dependent upon the expression of the CD3 on the T cells and the GP100 on the immune cells, and the on and off rates from binding to the CD3 and the GP100. So I think if you can take this information, it makes sense actually. Rituximab is dosed at different doses for different diseases. So it's a higher dose for chronic lymphocytic leukemia than non-Hodgkin's lymphoma, and it's due to the B cell load. So you're adjusting based upon effectively your expression of your target. And this work lends itself potentially to the future to bolting on multi-omics approaches, single-cell sequencing to better understand patients where they may be in terms of their expression levels, so that you could potentially design in a rational step-up dosing. This is an intravenous drug, so that should be possible potentially, and we could have treatment algorithms. So looking ahead, I'm going to just touch in the last 5 minutes, touch on can we be using artificial intelligence more to augment QSP? And of course we already are doing, and I'm not going to try and do an exhaustive review here, and it's a very complex field, but it's the concept of what we can do that's so important. We published a little narrative earlier this year in CPT-PSP where we considered AI approaches to augmenting clinical pharmacology, and we kind of put it into a Venn diagram where we would have an effect on improving efficiency of our clinical pharmacology decision-making and design, enabling more accurate predictions, and producing unbiased decision-making, which is critical. Some other ideas in terms of AI. I mean, we could be using it to understand the variability in response in CRS in examples 2 and 3. I think I've spoken about that quite extensively already. But as AI and machine learning matures, we may be able to build QSP models de novo through algorithms. So saving time on those models, model building, which can take months, years to put together. And finally, reversing translating QSP models to refine drug discovery. This is a little schematic from Lucy Hutchinson et al., again in CPT-PSP a number of years ago. And this is to sort of explain how we might do this. So integrating multiple data sources and models into machine learning models. So on the left-hand side, you have your prior knowledge and your selected data. You fit it, you derive your M&S modeling simulation parameters, then you predict your outcome, your time course and variability. Classic PK/PD. On the right-hand side, what we do is you take your M&S predictions, put them into an output, and then add in input from omics and imaging, and train a machine learning model. So you combine all of this together to get a predicted time course and variability based upon those characteristics, those additional covariates and an understanding of the patient disease status. So by establishing this link, we get a better understanding of the impact of variability, and we can use that for simulation purposes. I'm just going to skip that slide. So, do areas for improvement remain in systems pharmacology? This is not a rose-tinted presentation. It's meant to be a presentation of what we can do, but a reminder of the areas that we need to focus on as well. So, absolutely areas remain for improvement. Data quality is key, of course. Over-parameterization is an age-old problem. You end up with confidence intervals which effectively cover the graph if you're over-parameterized. How well do we understand the disease that we are purporting to model? How accurate is the diagnosis of the disease? Thinking particularly of neurological conditions, where it's quite subjective. The ability of the model to have an impact, you need to be building it early and testing assumptions with emerging data. If we're building it in phase 2, often it's too late. We end up doing a retrospective analysis when it was available to us with the glyphosins, but with others, we've missed the boat. And finally, communication of the work. I would encourage anyone interested to read the Gitschker and Carr manuscript in CPT-PSP last year about how to communicate with audiences, particularly when we're dealing with complex models which can intimidate people, and so they switch off. How do we make this or ensure that this area is digestible, understandable, and enjoyable for the audience. Finally, I think we do have a bright future if we remember the pitfalls, and we can link technologies to not least better describing physiological and pharmacological systems, better understanding and describing variability within that system, enabling better solutions to managing patient variability in response and safety, and yielding more informed dose optimization decisions at an earlier stage. And with that, I'd just like to thank you all for listening.
Thank you very much, Alex, for a great talk. Maybe I can start with a question, if I can. So you'll hear from the next talk that what we're dealing with in clinical practice are patients with multimorbidity, multiple diseases, and so on. But all quantitative systems pharmacology models I look at and in the literature are based on single diseases. Do you know of anybody who's actually starting to look at QSP models where you can actually start looking at multiple diseases, modeling those different diseases and the interaction between different diseases and how they affect response to therapy? I realize that's quite a difficult thing to do, but I think somebody needs to start doing this.
Yeah, thank you, Munir. It's a great question, and I know it was something I was thinking about in the context of the presentation. I'm sure people in the audience know if there are good examples in the literature in this area. I haven't seen one that I thought that I wanted to present on. But I do think it's an area that we really need to focus on. I guess maybe I'm being a bit conservative. I kind of think we still need to be ensured that we understand the variability and the disease characteristics of a single condition before we put it into the multimorbidities. But again, that may be something for artificial intelligence, that you can capture each of those models and bolt them together. That would be something that could be something for the future.
Okay, thank you.
Thanks. Yeah, I think we've neglected systems pharmacology in recent years in favor of target-driven pharmacology, and I welcome your talk very much. I think it's very useful. Recently, talking about AI, I recently reviewed the literature on AI and pharmacovigilance, and I was struck by the fact that the vast majority of papers in the field are published by computer scientists. And clinical pharmacologists have very little impact on this. There are some, but there's very little of it. And I think we need to be encouraging clinical pharmacologists to tool up in AI and become more involved. You mentioned communication. Are there any other ways we can do this?
I suppose, try and think of that concept, that question in the round. So in an academic situation, cross-collaboration across departments and disciplines. It would be the same within the pharmaceutical industry as well. Project teams should be working early on the concepts of QSP and integrating particularly computer science techniques which enable us to deal with these more complex systems to the concepts of basic pharmacology and clinical pharmacology. So I just encourage teamwork. I'm not quite sure how one could mandate that, but I think it would be the key to the success.
Okay, any more questions?
Yes.
Serge.
Hi, Munir. Serge Kremers, Columbia University, New York. Jeff, you actually, I think you actually opened my mind a bit, because were those papers that you reviewed, were those of bad quality? And might it be that we don't need any clinical pharmacologists for this? It's actually a question for you, Jeff.
I'll sit back and let Jeff answer.
Okay, thank you. So can I ask another question, please, Alex?
Please, Munir.
So SGLT2 inhibitors, and you showed them a modeling for that in terms of glycosuria, etc. But now there's sort of widening indications for SGLT2 inhibitors, particularly in heart failure. And every talk I listen to on the SGLT2 inhibitors in heart failure say they do not know the mode of action. Can your systems pharmacology models help us to understand the potential modes of actions in heart failure?
Yes, but I think we need to understand the basic pharmacology. So that may come to what you said about multimorbidities. as well, that if you could link the understanding of glucose changes to the propensity for a cardiovascular change. Although I don't think that's what it is. I think there's other sequelae as a consequence of the inhibition, which causes the benefits in cardiovascular disease. I don't see why that couldn't happen, but you've hit on the point that Duncan mentioned earlier, and I think about how many times I read a drug label, and the first sentence in section 12 in the label is the mechanism of action is not completely understood. And while that remains, QSP may be able to add insight, but it's not going to be, it won't necessarily unearth something that nobody ever knew. So I think that might be quite slow progress.
Okay, thank you. So last question here, please.
Thank you for a great talk, François from Lausanne, Switzerland. For clinician, how would you characterize the difference, the seminal difference between PBPK and QSP, as there is some overlap in the modeling approach?
Thank you. Thank you for the question. They're both effectively systems pharmacology approaches. The main thing is that what they're purporting to. So yeah, maybe we lump these together a bit too much. A PBPK approach is to understand the basics, the physiological basics which impact on the changes in exposure. And you could also extend that to things like prediction of drug interactions, which may change those physiological based consequences. The QSP, which I'm referring to principally here, is more about the understanding of the disease and the pharmacology and the exposure which may impact upon modulation of that pharmacology. So there's a slight difference between the two, but they come under the same umbrella of systems pharmacology approaches. Does that answer your question?
Yes, absolutely.
Thank you.
Thank you.
Thanks very much, Alex.
Thank you.
Thanks very much, Munir. I'm going to chair the second half of this. So we've been particularly focused on the earlier stages of drug development, and in the second half we're going to focus on those that are a little bit later. As has already been mentioned, we still basically develop therapeutics in single disease mode, but patients don't operate in single disease mode. Most patients have multimorbidity, and with that goes polypharmacy. And done well, that means that patients can have a great response. Done badly, it means that they have a huge burden of adverse effects and don't derive therapeutic benefit from that. So I'm delighted to introduce Dr. Chris Threapleton from St. George's, who is a specialist registrar on Clin Pharm and general internal medicine, who's going to talk to us about a systems approach and a patient focus on polypharmacy. Thanks very much.
Thank you for the introduction, and thank you to the organisers of WCP for inviting me to speak today. And I'd like to start my presentation with a confession, actually. I love my job. I'm a registrar in clinical pharmacology, but I never intended on going down that path. I've always been interested, ever since I graduated from medical school, in treating people with multiple complex long-term conditions and treating them in a holistic way, taking into account their own thoughts and values. So my path was to become a geriatrician. And it was only when I started working as a teaching fellow in clinical pharmacology in 2017 that I realized what potential clinical pharmacologists can have in the management of people with multiple complex long-term conditions. So I'd like to speak to you today about one aspect of that, polypharmacy, and the role that we might be able to play as clinical pharmacologists. I've no conflicts of interest to declare in this talk. So my talk is going to be divided broadly into 4 different areas. So first of all, I'll look at how polypharmacy is managed currently. I'll look at what factors drive polypharmacy. I'll then move on to explore what we might do as clinical pharmacologists in tackling this area. And I'll use our service at St. George's that we've set up as an example. And then finally, I'll reflect on where we might go as a profession next. So polypharmacy essentially means the use of multiple medicines, but there's no clear definition of exactly what that is. Typically, we think of polypharmacy as being around 5 or more medicines taken regularly. And around the world, between 12 and 20% of adults will take 5 or more medicines. You also may hear the term excessive polypharmacy. Again, no strict definition of this, but typically we think of this as being 10 or more medicines. Now, of course, taking multiple medicines is often appropriate. The more long-term conditions you have, it's more likely that you'll need to take multiple medicines to be able to manage those conditions appropriately. But as with all medicine, there is a balance between risks and benefits. And we know that polypharmacy can be inappropriate when those harms outweigh the benefits. Harms from polypharmacy come from a number of different sources, including adverse drug reactions, drug interactions, medication error. We heard yesterday that I think the costs of medication error around the world is something like $42 billion US. And also, more broadly speaking, patients often feel very burdened by the number of medicines they take. So we need to make sure that if we are going to be giving our patients lots of medicines, that they are the most appropriate ones for them to take, and they are more likely to cause harm, to cause good than harm. So how is polypharmacy managed currently? I think this is obviously important to look at if we are to see what we might be able to do in addition. So I'm going to use the UK as an example here. So polypharmacy is typically dealt with through medication reviews, and this might be simply just looking through someone's medical record to look at their diagnoses and prescriptions to see whether those prescriptions are appropriate. But guidelines are now stressing the importance of a structured medication review. So there's a nice guideline which advocates the use of a dedicated consultation between a clinician and a patient to discuss not only the medical perspective of the diagnoses and the prescriptions, but also the patient perspective, exploring what their values are, what their priorities are when it comes to treatments. Now, traditionally, this work has been carried out in primary care, typically by general practitioners, but with also, of course, with support and input from secondary care specialists such as geriatricians. But increasingly so, this work is being carried out by clinical pharmacologists. Certainly in the UK, there's been a big drive over the last few years for more clinical pharmacists to work in primary care in order to carry out structured medication reviews for people with polypharmacy. And you can see here this map of my patch in southwest London, and each of these coloured circles represents an individual GP practice. GPs are typically employed directly by the GP practice, whereas pharmacists are increasingly employed by small networks of GP practices, which are called primary care networks. And the aim of primary care networks is to share expertise and resources, and often pharmacists will work perhaps across a number of different GP practices, possibly spending about a day a week at each practice to review people with polypharmacy. So the landscape's changing, the workforce is changing, and there's been a big push to actually really address polypharmacy more directly with people with complex long-term conditions. So I think this raises a really important question here. How well prepared do healthcare professionals feel to be able to tackle polypharmacy and to tailor medicines to individuals based not only on their medical conditions but also on their own values? I conducted a survey of GPs and pharmacists and GP trainees across southwest London. So this went out to all people who worked in primary care. And I received responses from 71 GPs, GP trainees and pharmacists. I'm just going to show you some of the results here, because I think this is actually quite interesting. So here's a figure which shows the responses to various statements that I asked. So I gave statements and asked people to respond whether they agreed or disagreed. And this figure shows the percentage of people who agreed or strongly agreed with various statements relating to tailoring medicines to individuals. You can see in the dark blue, we have GPs, light blue GP trainees, and green for the pharmacists. And overall, actually, most people felt a sense of personal responsibility to tailor medicines to individuals with polypharmacy. Most people felt that they were able to explain medicines in a language that patients understand, and also felt able to make decisions with patients about what to do with their treatments. By and large, most GPs and pharmacists also felt they had the consultation skills to carry out structured medication reviews, but understandably less confidence from GP trainees. But here is where the problem lies, I think. So very few healthcare professionals felt they had sufficient time to be able to tackle the complexity of these patients. Lots of people felt they had problems with a lack of training, possibly, or a lack of clinical supervision. There was concern about access to secondary care for specialist advice. Lots of people felt they had perhaps not enough knowledge or skill in interpreting existing guidelines and evidence. And there was also a concern about a lack of confidence, again, particularly with GP trainees. So with this in mind, let's now look at the burden of polypharmacy. We've talked about the burden at an individual level, but where is polypharmacy the greatest? Perhaps there are areas that we as clinical pharmacologists might be able to focus on to try and improve what's already happening. So here's a figure showing individual GP practices. So this is the proportion of patients who take 8 or more prescriptions within each GP practice. This data comes from the polypharmacy prescribing comparators published by the NHS Business Services Authority, which has a number of different markers, and one of them is 8 or more prescriptions, which I use as my sort of surrogate for polypharmacy. And I've lined up the GP practices here from the one with the lowest level of polypharmacy to the practice with the highest level. And you can see there's a 5-fold difference in polypharmacy prevalence between different GP practices. So why might that be? How much of that might relate to possibly a lack of knowledge or skill at tackling polypharmacy? So maybe that might be something that we could do as clinical pharmacologists to improve on. Or possibly there are other factors at play here, possibly differences in demographics of the patients at the different GP practices that might be driving some of this variation. So the next step was to try and explain this variation. Why is it that certain GP practices have a higher proportion of patients with polypharmacy than others? In order to do this, I carried out a multiple linear regression model. And it was really important for me that I included factors in this model where there was an evidence basis. So are there factors that we already know drive polypharmacy? So the first step was to perform a systematic review and meta-analysis of existing research to see what was already out there. And I conducted a rapid review. So this is a review using methodology very similar to systematic reviews. It's slightly more abridged, and it can be done in a shorter time period, but also employs similar sort of rigorous methodology using guidelines published by Cochrane. And my search, once I'd whittled down the papers, I included 88 papers from 50 countries, which looked at a wide range of different factors that potentially might be associated with polypharmacy. Here's my meta-analysis of my maximally adjusted pre-calculated effect sizes. And you can see here are all the factors that are significantly independently associated with polypharmacy. So being older, so being middle-aged or older compared to being a young adult associated with significant polypharmacy, having lower socioeconomic status, having less education, currently or previously having smoked, being obese. And there's also a number of health factors which mostly relate to multimorbidity, so essentially having more long-term conditions, having lower self-rated health, being more frail, and also having been hospitalized recently. So with this in mind, knowing what factors are independently associated, I then needed to find factors at a GP practice level that might explain this variation. And there's a number of different databases out there which publish information for each GP practice. And so here are some of the databases I used. So for age, you can find out, for example, the proportion of people aged 65 and over per GP practice. I used the English indices of deprivation for my markers of socioeconomic status, and also the educational deprivation for my marker of education. And you can look at also other aspects, such as the percentage of people who smoke or who've smoked previously or who are obese. There's also a large national GP patient survey which goes out on a regular basis, asking patients a whole wide range of questions, including things like how many long-term conditions do you have syndromes which sort of indicate frailty. So I was able to use proxies here which might explain frailty, and also when people were last hospitalized. So I used data informed by my meta-analysis to go into my multiple linear regression to try and explain this variation in polypharmacy. But I wanted to go further than that and look at what aspects might need to be in place to tailor medicines to an individual. I mentioned that you can't simply follow guidelines. These patients are very complicated, and often you need to take into account their own thoughts and values. So I used information from a recent research project which produced a model called the Dexterous model, or Dexterous framework. So this was a large research project funded by the National Institute of Health Research, which asked the question, what needs to be in place in order to tailor medicines effectively and safely for an individual with polypharmacy? And this research looked at lots of existing publications and came up with 4 key factors that need to be in place. So in order to tailor medicines for an individual, we need to have access to high quality, consistent, contextual data about our patients. We need to know what their long-term conditions are, what their test results are, what their symptoms are. There needs to be a shared explanation and understanding between the clinician and the patient about what the issues are, what the potential goals might be for various treatments. There also needs to be trust between the clinician and the patient, that the clinician is working in the best interests of the patient. Trust that the clinician themselves feel able to tackle this issue and actually create effective change. But also trust in the process itself, that actually if you were to make a change to someone's medication, that hopefully they would come to more good than harm. And so that's an important aspect of that is continuity of care. And all of this needs to take place within a supportive infrastructure. Clinicians need to feel empowered and able to tackle polypharmacy for their individual patients. And they need to have the time and the resources to be able to do so. So, informed by this, I wanted to include information from the Dexterous framework to explain some of those, the variation in the prevalence of polypharmacy. Now, it was difficult finding data that might represent some of these factors, but I've used a few different databases again to create proxies that might explain this variation. So the general practice workforce data allows you to look at the number of GPs and pharmacists that work in individual GP practices, and you can look at the full-time equivalent of these professions per 1,000 patients. And this might give some idea of potentially the workload that GPs and pharmacists have within their individual practices. For shared explanation and trust, I went back to the GP patient survey I mentioned a second ago, which asks a whole range of questions to patients about their interactions with their health professionals. In terms of shared explanation, there are questions about feeling listened to, feeling you've been given enough time to talk about your problems, feeling your needs were met, and feeling you had an opportunity to share in decision-making about your medicines. And with regards to trust, questions about trust in the clinician, feeling they were treated with care, and that they were able to see the same clinician each time if they wanted to. And I used principal components analysis to reduce down the number of factors here to create a component that would be my proxy for shared explanation and a component for my proxy for trust. Unfortunately, I wasn't able to find any data at a GP practice level that might represent access to data. And finally, I thought it would be important to represent some idea of prescribing behaviour. Perhaps the difference in the polypharmacy levels between GP practices is not down to demographics or other factors, but down to simply some GPs or pharmacists giving out more medications, feeling more inclined to prescribe for certain conditions than others. And does that play a role in polypharmacy? Now, there was no existing research available which gave any sort of idea about how best to do that. So I had to go back to sort of mechanisms or to try and understand what might represent potentially poor prescribing. And I used the openprescribing.org website, which publishes the numbers of prescriptions of various different drugs within each different GP practice. And I picked 4 here that may represent some aspect of prescribing behaviour or potentially of poor prescribing. So I picked antibiotics, You'd imagine that maybe GP practices where there's a very high level of antibiotic prescribing. Now this is also, I should say, antibiotics here are the level was adjusted by age and sex. So potentially GP practices that are giving out more antibiotics may represent some overuse. Anxiolytics, hypnotics, and opioids may represent some, potentially some clinicians providing more pharmacological approach to problems when maybe a non-pharmacological approach might be more appropriate. And there's also a list published by NHS England of low priority treatments. So these are drugs where there's a really poor evidence basis and they shouldn't be prescribed. So things like cough syrup, for example. So again, I use principal component analysis to combine the rates of prescribing of these medicines per 1,000 patients to produce a proxy for prescribing behavior, I guess you'd say. So what did my linear regression model show? Well, here are the results, and I've highlighted the factors that were independently associated with the difference in polypharmacy between GP practices. So male sex, being older, having lower education and lower socioeconomic status were all significantly associated. But so was poor prescribing. So I think this is a really interesting point to raise here, and this is a potential role that clinical pharmacologists could play. We can see here that potential poor prescribing is a driver of polypharmacy in different GP practices. So perhaps that might be a role that we could play, upskilling people to improve the quality of prescribing and potentially drive down polypharmacy or certainly make it more appropriate. So let's now look at what clinical pharmacologists might do. There's a lot of research out there about interventions generally speaking and how they might, what outcomes they might result in terms of trying to make polypharmacy more appropriate. Unfortunately, there's very little looking specifically at the role of clinical pharmacologists. A lot of this, so these are all systematic reviews, a lot of this has been looking at other healthcare professionals. And there's mixed or contradictory evidence that interventions to improve polypharmacy have any impact on adverse drug reactions, on hospital admissions, health-related quality of life, and really no or very uncertain evidence that there's any effect on overall quality of life or mortality. But there is strong evidence that interventions can have some impact. Impact in terms of improving the appropriateness of medicines, reducing the number of medicines, and also improving symptom burden. So people feel better after having had their medicines changed. And also patients are very keen to have their polypharmacy addressed to make sure they are taking the right medicines. But I think there's a few real problems with existing research. As I mentioned before, there's very limited research looking specifically at the role of clinical pharmacologists. The research is very heterogeneous for a number of different reasons. Polypharmacy is a syndrome, it's not a disease process. So the people who have polypharmacy are often very different from each other, and it can be difficult to compare both within a study and between studies. The interventions in studies are also very different from each other. They're often quite poorly described in publications. It's difficult to know exactly what people have done. And often the interventions aren't based really on any kind of previous research or underlying theory. And outcomes are also, again, quite heterogeneous. And often have really quite broad measures such as mortality, which is unlikely to be affected by reviewing someone's medicines to make them more appropriate. There's also been a distinct lack of patient and public engagement in this field. We don't really know what patients feel about polypharmacy. What would be their priorities if we were to tackle this? So I'd like to now move on to talk about what we've been doing at St George's in London as potentially sort of a model that people could use to address polypharmacy. So we have set up a polypharmacy service run by clinical pharmacologists at St George's Hospital in southwest London. And the idea is that we would act as a sort of specialist hub within southwest London to upskill and support the existing work happening in primary care and also see some of the more complex patients ourselves. And we devised our service based upon the results of our survey and of our regression model. And you can see on the right-hand side here, there are three key aspects to our service. We have an advice and guidance service where we can respond to queries about complex prescribing decisions. And hopefully this will go some way to addressing that limited access to secondary care. We have regular multidisciplinary meetings where we invite GPs and pharmacists to bring cases of complex patients for discussion. And this will hopefully go some way at improving supervision for dealing with complex patients. There's also a big fear that was raised in the survey of, well, what if I make what if I make the wrong decision? What if my patient comes to harm? So hopefully having that discussion within a larger team can allay some of those fears. Hopefully it can improve confidence and also improve training. And we have carried out a number of workshops as well to upskill people in primary care to improve prescribing and people's ability to tackle polypharmacy. And we also have a polypharmacy clinic where we see some of the more complex patients ourselves. We have 40-minute appointment slots, so we have the time to really look at and address both the medical complexity and the complexity of the patient's own thoughts, values, and lifestyle. Now, I'd like to just show you now our approach to tackling polypharmacy as clinical pharmacologists. And I've got a case study here, and I'm not going to go into the clinical aspects of this case. Really, I'd just like to use this as a way to show what we might do as clinical pharmacologists if we had a patient come into our consultation room with polypharmacy. So here's a typical referral letter that we might get. So this patient here has a number of symptoms. She's been falling over frequently. She feels tired and is unsteady on her feet. And typically we would get a past medical history in chronological order and often an incredibly long drug history in alphabetical order. And it can be really challenging to try and work out what on earth to do with this, mess sometimes in the medical records. How do you line up the individual diagnoses with the prescriptions and work out what to do? This is really challenging and complex. So we have devised a structure to address this complexity and make it easier to come up with decisions with patients about what to do with their medicines. So this is the clinical pharmacology structured review. And this essentially is a table to structure your own thoughts as clinicians and then also use this for discussion with patients. You can see on the left-hand side here the different body systems, cardiovascular, respiratory, gastrointestinal. So rather than trying to line up individual diagnoses with prescriptions, simply put everything related to the cardiovascular system in that row. So you've got the diagnosis there for our patient. We've got test results that might explain possibly how severe diagnoses are or how well controlled they are. You can then add in the medicines that are related to cardiovascular disease more generally. And then at this point, you've got a clear idea of the medical perspective of this patient. What are the medical issues? What might you as a doctor want to do to address some problems? At this point, you can then take this to the patient and you can explain why they're on certain medicines already and explore their own concerns. What symptoms do they have? What priorities do they have that they would like to address in a medication review? And then together you can come up with some shared decisions about where to go next with their medicines. And all of our patients will receive a copy of this with their clinic letter as well, so they can see exactly what's happened, what decisions we've made. We developed the CPSR using anonymized GP data. So we conducted simulated medication reviews of 100 patients aged 65 to 84 who took on average around 10 medicines each. And we performed these medication reviews and gave recommendations of the sort of things that we would do if we were seeing that patient in clinic ourselves. We made, on average, around 5 recommendations for people's medicines. And we would recommend stopping around 1.7 medicines per patient and starting 0.7 medicines. Because I think it's also important to recognize that people with polypharmacy can also be undertreated. It's not just stopping medicines, but it's also important to think about medicines that should be started. But what was really interesting about this research was we also looked at the time since their last actual medication review in primary care, documented in their records. And we found that using our systems approach to tackle polypharmacy, we identified issues regardless of the time since their last medication review. So a patient could have had an actual medication review a month ago, but we made the same number of recommendations than we would have done if that patient had had a medication review not for a long time. So this indicates the possibility that clinical pharmacologists can provide an additional benefit to what's already happening in primary care using a structured systems approach. So what's been our progress so far? So we've set up our service, everything is up and running now. We've had 116 complex referrals about prescribing decisions via our software package Kinesis. We've held 40 multidisciplinary meetings to discuss complex patients and held 5 prescribing workshops for GPs and pharmacists. We've also had 149 referrals to our clinic as well, and still going strong. So let's look at the future. Where are we going to go with this project next? Well, I think in terms of the clinical pharmacology structured review, the next step will be to evaluate the performance of this in real life. Are we making the same number of recommendations and the same quality of recommendations for people when we see them in real life medication reviews compared to those simulated reviews? It's also important to engage patients and the public, and it'd be important that, that patients are involved in further development of this table, so they're getting out of it what they would like to get out of it, and that they understand exactly what's going on with the medication review. And in terms of evaluation of our service, it's important to evaluate not just outcomes, but also the processes of the service itself. You know, who's referring, what sort of people are being referred to our service, how is that working? Because if we aren't getting engagement from primary care, then the service will fail. So we need to make sure that the service is successful, and then also look at the outcomes that we might be able to get. And not just outcomes in terms of the number of changes to medicines, but also more patient-focused outcomes. We're probably not going to make any difference to mortality, but we hopefully would make some improvements to symptoms. And I think the way to go with research into complex interventions like this is to focus on what patients would like to achieve from an intervention. So our patients are all very different from each other, but perhaps one patient would like to improve a particular symptom. Maybe someone else would like to be a bit more mobile by feeling a bit less dizzy, or maybe someone else just wants to take fewer tablets. We ask these questions anyway in our consultations, and we can then, one way of evaluating our progress is to see how well we achieve those goals that were set with our individual patients, rather than using some overall umbrella outcome for all people. I think it's also important as clinical pharmacologists that if we take this on, that we actually embed ourselves into existing structures. And so that's working with GPs and pharmacists, but also with other structures such as the Academic Health Science Network. So we've been working with our local AHSN group recently. They have a national polypharmacy program to try and both deal with people with polypharmacy and reduce inappropriate polypharmacy in the first place. And I think this is a real opportunity for us to work with the AHSN to become more involved in management of people with polypharmacy. So where might we go next as a profession more generally? So clinical pharmacology is a very, I think, exciting and varied career, and we'll always have roles in education, in medicines policy, and very important roles within research and clinical work. But I think there's a real potential for us to include polypharmacy within our umbrella of the various things that we do. Certainly at St George's, by having an outpatient service, we've really raised the profile of clinical pharmacology, both in primary care and also within our hospital as well. People are now more aware of what it is that we do and who clinical pharmacologists are. And there's the potential to add in other things into the service as well. Pharmacogenomics is a hot topic at the moment and would be increasingly relevant in clinical practice. So having a clinical pharmacology outpatient service gives you the potential to add in pharmacogenomics. Other aspects might include things like allergy delabeling, potentially bringing people in for outpatient appointments, people who are in clinical trials. But also I think another important point to raise here, I mentioned at the very start, I never intended on becoming a clinical pharmacologist, but I'm very glad that I have ended up as one. But there's a lot of people out there who don't know what clinical pharmacologists are. And I think if we as clinical pharmacologists champion our role within the management of complex patients with polypharmacy, we really have a potential to attract many more people into the specialty. So that's the end of my talk. I've got a long list of acknowledgements here. So this research was part of my PhD that I hope to finish in the next couple of months. But I really couldn't have done this work without the support and guidance from lots of people here. I'd like to, if I may, just mention a few people. So Professor Emma Baker, who's in the audience, has been a terrific support and guide over the last few years, particularly with this PhD, alongside Professors Tess Harris and Joanne Reeve, who was the author of the research project which produced the dexterous framework. I'd also like to say particular thanks to Katie Feather, Michelle Sylvester and Rhea Shah, who are in the audience, who are an integral part of the polypharmacy service at St George's and who've been really supportive with my research as well, so thank you very much. They're my references, and I'd be very happy to take any questions.
Thanks very much, Chris. We've got time for a couple of questions. If I could just start, do you think there's an opportunity to use some of the systematic approaches that you've got to sort of formally look at benefit risk of some of the sort of more common combinations that you're seeing, to actually see whether in the real world the combination of these things really delivers what we think we're doing when we're thinking of them in a sort of mono mode?
Yeah, I think that's definitely the case. We're potentially creating a very large database of information about complex patients who take various medicines, and potentially we'll have an opportunity to look at outcomes as well. We do use things like number needed to treat and number needed to harm for our patients occasionally when we discuss individual patients and treatments, but obviously that doesn't take into account, as you said, multimorbidity or being on lots of different medications. And I think there's a real role that of within polypharmacy that we could look at this, yeah.
Great. Robin, I think you've got a question.
Yes, thank you very much, Chris, for that. You showed that poor prescribing amongst general practitioners is one of the drivers for polypharmacy. I know that at George's, the medical undergraduates get excellent training in clinical pharmacology. I just wondered whether you'd looked at the duration of the effect of good undergraduate teaching, or to put it in another way, do you know where your GP is qualified?
That's a really interesting question. No, I don't is the short answer. It would actually be quite interesting. So you can, for example, look at, I think within the GP practice workforce database, the time that GPs have been qualified, I think. So that might be something that we could look at. Within our survey, we asked people, the GPs, the pharmacists, how long they'd been working in healthcare for. Unfortunately, we weren't able to map those responses directly to individual GP practices, but it would be interesting to see. Certainly from the qualitative responses we had from the survey, people often felt that, yeah, as you say, they have good quality training when they're at university, but then very little since. And actually now, especially if you've been in healthcare for a long time, things are changing, patients are more complex, and I think it can be really challenging. And certainly everyone's been very supportive in primary care of our service, because lots of people will say, I've got a list of 10, 20 patients I have no idea what to do with. And I think actually there's the potential to sort of work with people to try and improve that.
Thanks.
Microphone 1.
Thanks. Hi, Kelly Charles, University of Sydney. Really fascinating work, Chris. Great PhD to come. I guess I'm interested, you presented very early that these GP practices or primary care networks have a pharmacist or pharmacists attached, and I'm wondering, how does that fit within your model of how clinical pharmacologists can interact with polypharmacy? Are you seeing the ones that don't necessarily go, the cases that go to the pharmacist within that small network, or is there any way of finding out from the ones that have very high prescribing of polypharmacy, are they utilising their pharmacists in their network appropriately? And are you seeing almost like a surrogate for lack of integration of interprofessional care?
Yeah, I think that's a really interesting point, because I think pharmacists are being used in different ways in primary care, and one of the common complaints we have is that a pharmacist may be brought in to carry out structured medication reviews, but instead all they end up doing is medicines reconciliation and kind of more kind of routine tasks. Certainly there's no relationship between full-time equivalent pharmacists in primary care and the prevalence of polypharmacy, but perhaps there's some effect on exactly what the pharmacists are doing in GP practices. Certainly it seems now that GPs simply do not have enough time to deal with the complexity and the number of patients that they have to deal with. And a lot of the polypharmacy work is now going to pharmacists. And the qualitative analysis that I've not presented here, GPs are really grateful for the support of pharmacists. But often I think perhaps pharmacists may feel that they don't have the confidence or skills in dealing with the complex patients that GPs refer. And so most of our interaction in primary care is actually with pharmacists. We can't do the majority of this work as clinical pharmacologists. We are, certainly in the UK, a really small specialty. So our role has to be in supporting what's already happening, upskilling, the hundreds of pharmacists and GPs, and seeing perhaps some of the more complex patients.
I guess I had a follow-up question then. How many people in your clinical pharmacology service? Because I think that is a context that's very different. I mean, in Australia, most of our big tertiary hospitals may have 1 or 2 clinical pharmacologists for a full network. And this type of model may not necessarily scale if you don't have sufficient specialists in the field.
Yeah, I think that's probably quite similar to the UK, actually. I mean, I think certainly in London, we're quite lucky. We have probably a greater number of clinical pharmacologists than a lot of other cities and more rural areas in the UK. Currently at St George's, we have 3 consultants. A number more consultants, but 3 involved in the polypharmacy service. We have 2 registrars. I'm currently the only registrar within the service, and we have two fellows as well. So I guess 3 junior doctors and 3 consultants. And I think, but yes, certainly this at the moment couldn't be done in areas where there are less resources. But this sort of service might be used as a business case potentially. If we're able to see that we are improving outcomes, this could be used as a model to then say that we do need more clinical pharmacologists in other areas.
Excellent. Thanks, Chris.
Thanks. I do want everyone to have a chance to answer their questions, but if we could keep them brief. Jeff, please.
Yeah, thanks, Chris. I see not a few patients. Give the history to the clinical pharmacist. The drugs are prescribed. They get adverse reactions. In one case, the patient died from drug toxicity. Why? They take the prescription to the chemist, get the tablets and put them in the cabinet, never take them in the first place. So how do you tackle this problem of poor adherence in patients who appear to be taking polypharmacy and aren't, and how do you measure adherence?
I think that's a big issue, and certainly the more prescriptions a patient has, the more likely they are to not take their medicines as prescribed. And I think a big aspect of this is actually patients maybe not feeling that they are being listened to, or their issues are being dealt with appropriately, and that they are maybe being fobbed off or being given medicines and they don't feel able to say to their healthcare professional that they're not working for them, or they don't know why they're taking these medicines. So one of our roles is around first of all, championing the patient's priorities. What is it that they want to get out of their medicines? Trying to make it clear that we are on their side. And then having an open, honest discussion about what they're currently taking. And then also educating the patient. And I think the clinical pharmacology structured review has the potential to help to educate patients about why they're on certain medicines, and what they're for, and which ones they should take and which ones they may be less important.
So we're going to have to make this the last question. Number 4.
Thank you, Chris. Jean Mernion from University of Sydney as well. In a number of the deprescribing sessions this week, we've heard about scales that people use to identify particularly high-risk combinations of medicines, such as the Drug Burden Index or the Beers criteria. They predominantly identify medicines with high anticholinergic load or sedative hypnotic loads. Are you using any scales like this in your work to identify particularly risky combinations?
Yeah, we do. Yeah, so we certainly use the anticholinergic burden score on a regular basis. I think it sort of depends on the patient. We don't have a strict system of what we use. We look at the patient, look at their phenotype, their diagnoses and medicines, and then decide what might be most appropriate to look at. Potentially looking at something like anticholinergic burden score, risk of bleeding, that sort of thing.
Thanks very much. Thanks very much, Chris. Really great talk, and thank you all for all your questions. So our final speaker of this morning is Dr. Julie Bullock, who's Senior Vice President of Integrated Drug Development at Certara. She's now working in industry, but has a background working for the FDA. Clinical pharmacology, as we've heard, is an area that does play a really crucial role in the decision to whether to license a medicine or extend its license in many ways. And we've also seen that it's a discipline that lends itself to mathematical and modelling approaches. But these are often not always used, and I think we're going to hear now from Julie about how clinical pharmacology information can be used in a much more novel and imaginative way to support regulatory decision-making. Julie.
Great, thank you, and thank you for the organizers for inviting me here to speak today. It's a pleasure to talk about the regulatory applications of clinical pharmacology. I will disclaim that this is a very FDA-focused presentation because that's where I was trained and come from. I have interactions with the Europeans, but I find the FDA, since it's a singular regulation entity, a lot easier to digest and take learnings from. So today I'll go through a little bit of how the FDA reviews and approaches clinical pharmacology for applications, as well as some of their research, which I think is really important. We'll go into bracketing, which I find to be the pillar of clinical pharmacology and model-informed approaches. And then we'll go through some other new and notable approaches within the space, and then I'll conclude with challenges. So every year the FDA issues a review of the year prior, and it usually comes with a lot of really great information about that year's approvals and what sort of scientific improvements were communicated throughout those approvals and through the research that's being conducted with the Office of Clinical Pharmacology. And this is an excerpt of some of the information that came out. I don't expect you to read all of this, but I have highlighted here in yellow for each one of these areas how pivotal model-informed drug development and data analysis, including population PK, exposure response, QSP, pharmacogenomics, PBPK, all come into each one of these areas, which are, I think, incredibly important for what we do as clinical pharmacologists, which is optimize doses for patients, mitigate risks for these patients, expanding treatment options for our children and adolescents, influencing development and trial design, as well as assessing genetic factors. And all of these are being researched not by conducting additional studies, but by using the data that we're currently collecting in a way that allows us to inform decisions. So for those of you that haven't looked at the recent approval, and the format has changed a slight bit, but for the most part, the reviewers at the agency answer four questions, which is like, that's not very many questions. But if you start to drill down into each one of these, we're really using clinical pharmacology to decide whether or not the available data provided pivotal or supportive evidence for effectiveness. And I'll come back to that pivotal component a little bit later. We obviously focused a lot on dosing regimen for both the general population as well as subpopulations. And then, of course, the food-drug or drug-drug interactions is something else that we focus on. And PK/PD, pharmacometrics, all of these modeling analyses are integral to answer these questions. So there was a paper published in 2001 by my former colleagues that goes into, how we are using clinical pharmacology and MIDD and the current status and the emerging trends. And a lot of this, this cartoon here on the left, I think is a really great way of how we take all these little subcomponents of clinical pharmacology and put them together, assessing both pharmacokinetics, dynamics, effectiveness, and safety in order to hopefully inform an actionable product labeling. But then we can also do it a little bit differently, where we take the input here on the right and use all these different MIDD components to create output that can also be used to help not only optimize dose, but help us with clinical trial designs and informed decision making. So this cartoon just goes through the I guess the different landscapes and timings of when you would use all these different model-informed approaches. I mean, granted, it doesn't mean that you can't use PBPK in later stage or earlier stage, or exposure response in earlier stage. However, for the most part, in general, with no variability shown, obviously, this is where you would potentially utilize all these different analyses tools. And at the bottom are the decisions that really need to be made during these different phases of development. If we were to bucket everything into certain phases, depending upon where you are in development, though, some of the phase blurring is definitely starting to occur. So I'm going to talk a little bit about bracketing, which I think is a really important component of clinical pharmacology and model-informed development. And it allows us to move beyond this precision exposure matching. So when I say precision exposure matching, I basically mean bioequivalence. It means making an adjustment on pharmacokinetics alone and not understanding really the inherent variability of the exposure response for both dynamics as well as safety and efficacy, instead of making a decision that's based on all of these things with equal weights. So in this cartoon on the left, we have an assumption that as we increase dose, which would be, on the x-axis, or it could be exposure, we increase efficacy and we increase toxicity. Now, I come from the world of cancer drug development. This is what we think for almost all cytotoxics. As we increase efficacy, we are willing, increase dose or exposure, we are willing to accept the increase of toxicity, and we eventually have to choose a dose that is the most tolerable, so that we don't kill patients at the same time as hopefully curing their cancer. But what we find in most areas actually is what you see on the right. So as you increase dose or exposure, your efficacy is eventually going to plateau, but your toxicity more than likely is going to continue to increase. Even in the oncology space, if we've moved from a more targeted approach, we're starting to see more of what I've cartooned here on the right side, versus assuming that on the left we should just be pushing dose for all patients. So bracketing is critical to understand whether or not we're dealing with an asset that has a profile that's here on the left versus one that's on the right. We would approach the development and what we do about dose very differently for each one of these profiles. So the benefits of bracketing, I think, are not well understood. And how we can use bracketing in development programs. So I'll start with actually the cartoon on the right. So just, what is bracketing? It's basically, showing your efficacy as well as your safety threshold. In this cartoon, I have used the red dot to show the optimal or max response. Any increase in dose or exposure, at least for a little bit until we get to a maximum tolerated dose, would be okay, but it's not going to provide us with any additional efficacy, but it could potentially give us additional safety issues. But we can use this bracketing, we can use this knowledge of what the efficacy threshold and the safety threshold to help us not only with dose finding, but also regimen changes, formulation changes, intrinsic and extrinsic modifications for dose, and supportive evidence of efficacy. In an era where you don't have an understanding of this bracket, anything that you do for these things that are in this yellow box would be bound by bioequivalence criteria. So you would have to match the exposures of your asset. You would not have any ability to justify why higher would be okay or lower would also be okay. So having this bracket established for your asset definitely allows you, more room to move about your cabin, so to speak. So for those of us that are, as clinical pharmacologists, we should know that bracketing is a very common use to establish the phase 3 dose. And in most therapeutic areas outside of oncology, we have nice dose ranging studies that might have dynamic markers or biomarkers, maybe other clinical markers, not necessarily clinical endpoints, but across a range of doses that gives us some understanding as to what dose or doses we would want to take into phase 3. But there are common limitations seen in these dose-ranging trials, and I've probably seen every single one of these throughout my career, as I reviewed many assets. But, you know, the dose range evaluated could be limited or have, you know, just incredibly large gaps in exposure coverage. And of course, in those instances, it's always going to happen that the high dose is too toxic, but the mid dose doesn't show efficacy. And so they're left in this world of what would he have gotten if we would have studied something that was in the middle of that gap. And so that's definitely something I see quite a bit of. A lot of people just don't want to collect pharmacokinetics. We have not enough duration follow-up. I understand, you know, the first speaker spoke about having to get in and get out of this really quickly, but if we don't follow for a meaningful amount of time, we've just done a dose ranging study that hasn't provided us any information on what dosing would potentially be best for phase 3, especially if there are time to event or duration of response sort of implications into our decision making. Relevant clinical endpoints are not evaluated. This is relatively common because we want to use a biomarker. We want to use something that informs decision quickly. And then sometimes we don't have good control arms, or there's small ends within this. But I think, you know, while we might take shortcuts in hopefully one of these sub-bullets here, hopefully we're not taking shortcuts in all of them, right? So, you know, maybe we can have a small n, but we collect pharmacokinetics in all patients. Maybe we don't include a control arm, but we include more patients in our dose ranging trial. So there's definitely some balance and give and take here. I'm not expecting perfect phase 2 trials, but hopefully we don't see gaps in every single one of these limitations that I have. So for those of you that are struggling with potentially designing a robust phase 2 trial, I would like to just make you aware that a robust phase 2 trial can actually be one of the adequately well-controlled trials for evidence to support approval, at least for the FDA side of the world. This is necessarily buried in about 4 different guidances and was definitely spoken very clearly by Robert Temple in 2015 at a Brookings meeting on clinical pharmacology, that instead of doing 2 phase 3 trials that are basically identical trials and identical patient populations with identical doses, that he would rather see a phase 3 trial and a robust dose ranging trial, because the dose ranging trial usually would give us more information about the dose response, which is incredibly important. So these dose comparison or dose ranging information, if conducted appropriately, I have to stress that, can be used as supportive evidence of efficacy, because it provides not only the pharmacological evidence that we need, but also the clinical evidence across a dose range and supports dose justification. So data requirements. You know, we obviously need pharmacokinetic samples in all studies. So I get it a lot. I get this question at least once a week. How many patients do I need for pharmacokinetics? And I then turn back to my medical colleagues and say, how many patients should we sample for the primary endpoint? They usually look at me strangely, and I'm like, you wouldn't subset your primary endpoint of a phase 3 study. You wouldn't collect only some patients' data in phase 3. You're going to get your primary endpoint on all patients. So why are we shortchanging the pharmacokinetics? We don't monitor safety in a subset. We don't monitor efficacy in the subset. So we need to be a little bit stronger here. I realize there's operational issues with pharmacokinetic sampling in later stages of development. And I think that we should be able to use our knowledge to overcome some of this, but understanding that even if we get all subjects with sampling, we're more than likely, because of the operational challenges, going to come back with a subset of data in the first place. But what you don't want is a subset of a subset, and that's usually what ends up happening. Healthy volunteers and patient population of interest for definitely all those pharmacokinetic studies as well. Pharmacodynamics, if applicable, we would like to have supportive pharmacology, biomarkers, surrogates. You know, obviously we shouldn't be collecting the kitchen sink, but if we feel that we need to do that in order to understand the asset to find how this drug is working, then by all means we should look at those things. And we can use other mechanisms such as AI to analyze these to see what, if anything, is significant. Safety across the dose range is critical to establish that bracket, as well as efficacy. And of course, it comes back to how we analyze this data, which would be through modeling, both population PK and exposure response. So I'm going to go through a couple of case examples relatively quickly because we're running a little bit behind. I might just go through two, I believe, just to showcase how this could potentially be used. So the first one is asciminib, so I'm not going to go through, but it's a drug for chronic myelogenous leukemia that was labeled both as an 80 milligram dose as well as a 40 milligram BID dose. So the phase 3 trial was actually conducted with the 40 milligrams BID. But when I was reading the label, I was like, well, how did they get 80 if the phase 3 trial was conducted with 40? So I went back into the review and looked at their dose ranging trial, which was relatively robust and conducted in with a Bayesian design, so not inherently randomized, but also semi-randomized. And they had nice dose ranging data across not only different regimens, but different doses. I have here boxed out doses that were expanded in both wild type patients and patients with T315I. This is actually a really great case, and they took different doses in these different populations because of the pharmacology informing dose selection, that the patients with the T315I needed higher doses and higher exposures to elicit response, and that came out of the animal data. So based on this expansion, they decided to take 40 milligrams BID into development, 200 for the T315I. But based on exposure response modeling that was conducted after the study was completed, the agency allowed them to also get 80 milligrams QD in label, which I think is the first time I've seen the agency do something like this. Because of the available data and the convincing exposure response relationship. So not only did this allow them to get an additional dose in their label that wasn't studied, which is technically more patient friendly than a twice daily dosing, they were also able to use this exposure response, this bracket, so to speak, to justify no dose adjustments for mild or severe hepatic impairment or renal impairment, even though there was an increase in pharmacokinetics seen with those studies. And they also have no dose adjustments for drug interactions unless the patient is at the highest dose for the T315I. This definitely simplifies the ability to treat a patient, monitor a patient, and dose a patient when you have this ability to use your vast data that you have across a dose range for labeling. So it definitely pays back. Otherwise, we would have had to make all these dose adjustments and all these edits in the label for all these different things that were modulating exposure, which isn't necessarily, it wasn't needed based on the exposure response. I'm going to skip through inclisiran, but this is also a really interesting, I'll go straight to this one because this was based on the renal impairment study saw significant pharmacokinetic increases, but because they also looked at biomarkers and LDL in the phase 3 study, they saw that their patients that they had higher exposures with pharmacokinetics had the same amount of reductions in LDL-C. So in this instance, if they wouldn't have had that data on that biomarker, the LDL, we would have made an actual dose reduction based on pharmacokinetics alone and potentially lost efficacy in these patients. So there was no correlation between the pharmacokinetics and the biomarker at hand that was really critical for clinical benefit. So we have to be careful here and really understand not only PK/PD, but PK safety and PK efficacy as we go through our dose and dose recommendations. So some other new and notable approaches that are coming out of the industries, or the FDA, the role of pharmacodynamic biomarkers in biosimilar development was the headline here, but I have in the yellow box down here is, can we use pharmacodynamic similarity to approve formulation or manufacturing changes for 351A biologics? And for those of you that aren't in the regulatory speak, the 351A biologics would be a drug that's not a biosimilar, but is approved as a BLA. So these drugs might go through manufacturing, cell line changes, scale up post-approvals, just like our small molecules, but if they fail a biosimilarity exercise, they can't launch those new manufacturing changes. And so how can we use some of these biosimilar-like learnings, and the agency has finally moved to a place where they're using pharmacodynamic markers in biosimilar development and apply them to new chemical entity biologics. And this actually was done in 2022 with teplizumab, which I won't go through, but it's an interesting case study in a manufacturing change that was done after the phase 3 trial, and the comparability study failed. They got an initial complete response letter from the agency, and then came back with a model-informed approach that used not only pharmacokinetics but pharmacodynamics to provide supportive evidence that this new manufacturing was not different clinically than the manufacturer that was used in the phase 3 trial. So this was the first time this has been done. And the FDA review is actually really interesting to read as you watch them kind of go through this journey of how they could potentially use the totality of data here to get this drug on the market for patients. Diversity in clinical trials is another very hot topic at the agency. Definitely looking to, this is mostly focused on ethnicity, but I think it also gets back to race and our age as well as gender. And diversity plans are being required, and we're starting to see complete response letters for studies that are conducted ex-US with little to no US ethnic distributions. You know, if you think about a more diverse phase 3 study, and then you have the pharmacokinetics and the biomarkers and all of this stuff collected along the way, you think about what we could potentially use, POP-PK and exposure response and some of our other modeling techniques that we have at our ability with a more diverse population, definitely ripe for opportunity. And I can't go without talking about machine learning and AI. I just want to talk about the different types of approaches that the agency is looking at with regards to how AI could be used in submissions. And it's definitely being thought of for both outcome predictions all the way to trying to design drug toxicity predictions and post-marketing surveillance. So it's really kind of, it's a little all over the place right now with regards to how AI is going to be used for regulatory decision making, but it's definitely on the forefront of their minds. So I'll wrap up now. So I think hopefully you have a good understanding of how we can use model-informed, or how the agency uses and how you can use model-informed drug development for decision making. And I do think that model-informed development will be a continued driver of innovation for health authorities for years to come. But we have a fair amount of challenges ahead of us. I think we've, you know, like as Alex's talk mentioned, you know, the complexity of some of these quantitative models and the complexity of some of these PK/PD models definitely demand more on the workforce. We need more complex thinkers. We need people that are good at math as well as pharmacology as well as healthcare in order to put those together. And coming with these people that have that, you know, all of this stuff together is incredibly, it's very difficult. So as a community, we need to do better with training. Communication is so important. Most people don't understand our mathematical magical models. And we get up there and we put equations up and people just shut down. So we need to be able to, while understanding the details, we need to get up out of the weeds and be able to say, this is what this model is telling me, and this is how it's implying that we change our development course, or this is how I'm going to treat this patient. So we can't focus on the details. We need to, you know, occasionally come up for those physicians that need it. We need to put faith in the totality of data, and I'm not just saying that you as developers do, I'm saying that the agency does as well. And I think that teplizumab was a great example of, you know, seeing pharmacokinetics, pharmacodynamics, you know, kind of overlap, and then they felt more comfortable about seeing both of these things work together to make the extrapolation that the clinical efficacy would be the same. I will mention that regulatory lag and the fact that we don't publicly promote applied cases. None of you would have probably known about the teplizumab case unless I came up here and talked about it, because it's not something that we discuss frequently. The company's not publishing it. It's not presented at conferences. You know, you have to, but we all can't read through all these summary basis of approvals in order to learn from every single thing. So I think that being able to come up and promote these applied cases of clinical pharmacology and model-informed development helps people believe that it can be used to streamline development, but it also helps, there is going to be a lag, right? So the drugs that were developed that are approved today were developed 5 years ago, and so we can't just continually use the cases that we see in front of us. We have to, believe that things are happening in the IND stage that aren't public as well. And lastly, these approaches must be global. So we can't, if the FDA is going to do some of this, but the EMA is not, it's not going to work, because you have to develop your drug for the lowest common denominator and the most conservative regulatory agency. Otherwise, none of these approaches are going to take off. So we have to make sure that there needs to be more integration between our health authorities from a global perspective, and that can be really difficult. I think the agency does a really good at trying to use some of these new tools and apply them to their decision making, but I'm not 100% sure how that's being done within Europe, because there is definitely a lag, even just in PBPK, how those agencies are utilizing those two different tools. So I'll stop there, and happy to have any questions.
Thank you very much. Some really important insights there, how not every question in drug development requires its own standalone randomized controlled trial. Can I ask you a question in terms of, these are really important and powerful approaches, but my impression is the uptake of them by industry is still pretty patchy. Yeah, what's your view?
Yes, it is. And I think, when we're dealing with probability of success and risk-based, which I usually provide all clients when I put together these types of approaches, there's always risk with using a bracketing approach. There will always be that. Whereas, there's no risk with regards to going and getting the clinical data and showing it. But I think that in some instances, the risk is worth it, because the study to collect the data that you need from a clinical perspective can almost be unfeasible. So what we have to do is use these if you went to the teplizumab, you're like, okay, well, if their options were to do this model-informed approach or conduct another clinical trial with the new formulation. But the indication is for prevention of type 1 diabetes in children. How long do you think that clinical trial is going to take? 10, 20 years? They didn't have 10 or 20 years of investment. They had no choice but to take this model-informed approach. So those are the assets that are going to use these things, but I think that we could still leverage it for other assets. I didn't present this example, but it was used for a drug, a complement inhibitor that was developed by Alexion. They used a bracketing approach for their sub-Q switch. Now, they could have easily conducted an efficacy study, because granted, it's an orphan disease. It would have taken a little bit longer. It's not 5 to 20 years long. But they still took the risk. So I think that people that are willing to take the risk are going to see rewards. But some people just prefer zero-based risk approaches, and so they conduct the studies. Yeah.
Well, can I thank you and thank all our speakers, and Sir Munir for chairing this session, and all of you in the audience for your lively participation in the symposium this morning. So thank you to all our speakers, and have a good lunch.